Howdy, my name is Michael Davis. And today we'll be talking about how royal actin induces queen development through an EGFR-mediated signaling pathway. And I'll explain much more what this means in a bit. To start off with an introduction, give a little background information about Apis mellifera. This is the Western European honeybee, and it is known to play a prominent role in pollination, contributing both to plant biodiversity and the quality of many vegetables and fruit products. As a whole, honeybee colonies consist of many different castes, including queens, drones, workers, and brood. Today, we'll be specifically looking at the caste queens and workers and what differentiates the two. As for caste differentiation, there are two genetically identical female castes, the queen and the worker class. Both castes are genetically identical but exhibit different phenotypic expression due to epigenetic regulation. This regulation is caused by the differing diets and the ingestion of royal honey, which will be the focus of today. Queens have a much larger body size and specialized anatomy. They also have a much shorter developmental time than workers, as well as a much longer lifespan. I mean, this makes sense as queens are responsible for laying thousands of eggs, while workers rear young larvae and gather nectar. When larvae are nourished with royal jelly, which is secreted by workers, they differentiate into queens. Royal jelly contains specific factors that determine caste differentiation, but the exact components and their corresponding mechanisms were previously unidentified. As for more on royal jelly, it is a nutrient-rich substance, much more so than worker jelly, and it contains an increased quantity of sugars as well as more royal actin, which we'll touch on later. Royal jelly induces or elevates the activity of several different pathways in queens as shown, which include the insulin IGF-1 pathway, EGFR, mTOR, and JH or juvenile hormone pathway. While the upregulation of these pathways through the respective mechanisms all play a role in caste differentiation, I will specifically focus on royal actin and its effects on the genes involved in caste differentiation, as this substance has previously been shown to induce queen formation or queen-like qualities. So the question today we'll be answering is, what is the effect of royal actin on the genes involved in caste differentiation? In order to identify which factors that induce caste differentiation in the honeybee, as well as their mechanisms that drive this developmental pathway, the components of royal jelly were analyzed and their effects on individual honeybees were investigated. To start off, the investigators showed that larvae reared with royal jelly stored for 40 degrees Celsius for 7 days showed increased developmental times, decreased adult body weights, and decreased ovary size compared with larvae fed with fresh royal jelly, which essentially means they're losing the queen-like development. And so long-term storage of royal jelly at high temperature decreases the biological activity of royal jelly for queen differentiation. And so on the other end, the more extreme end of the spectrum, adult females reared with royal jelly for 30 days at this temperature develop into full worker morphotype. And so essentially these are the two ends of the spectrum that we'll be looking at today. And so knowing that the queen-inducing effects of royal jelly decrease over time as the jelly degrades, we can identify the various levels of royal jelly components over time and see if they correspond to the levels of queen development observed. And so first, to look at these components, the investigator examined vitamins, carbohydrates, and fatty acids and compared their differences over these different temperatures. And overall, there were no significant differences throughout storage. So next up was proteins. Proteins in royal jelly stored were analyzed, and 2 proteins were shown to degrade during storage due to the temperature, a 450 kilodalton protein and a 57 kilodalton protein, which is called royal actin. And those are the 2 we're going to be looking at today. And so this essentially lays the groundwork for this first figure, where we essentially have several groups being analyzed for their effect on caste differentiation. As a control, we're looking at larvae reared with fresh royal jelly. And then on the other end of the spectrum, we have that royal jelly that's been degraded for 30 days at 40 degrees Celsius, or the 4330D RJ. Next, we have the same degraded royal jelly stored for 30 days, but it also contains different components. So one of them will be casein, which is a nutrient control that functions to provide the individual with energy, the 450 kilodalton protein we mentioned, and the royal actin. There's also a group called the E royal actin, which is essentially just a recombinant royal actin expressed by E. coli and then purified. And so to compare the degree of queen development induced in these individual larvae, the investigator looked at developmental time, weight at adult emergence, and ovary size. And then here we can also see a picture of the final adult size. And so the main takeaways from this figure are that the 450 kilodalton protein in casein did not change the final adult size, developmental time, or ovary size, while in contrast, royal actin did on all of these. We'll also note that larvae reared with recombinant royal actin showed very similar results to the original royal actin, and overall indicating royal actin had a significant morphological effect on caste differentiation, making it a major factor. Next up, to analyze the effects of royal jelly on model organism Drosophila and their larvae. And so essentially, these flies are a well-studied model organism in gene regulation and epigenetics. So we look to confirm the queen-inducing effects we saw in bees in this other model organism. And so we were looking to induce similar morphological and physiological changes in Drosophila, similar to those induced in honeybees. And so in part A, we have the compared body size of female adult flies reared in casein, a control substance, and royal jelly, which we can see a very big difference. In B through D, it's very similar to figure 1, where the experimental groups consist of flies reared with control medium, casein medium, royal jelly medium, and then the royal jelly stored for 30 days at 40 degrees Celsius, possessing either casein, the 450 kilodalton protein, or royal actin. And so once again, to look to analyze the morphological differences, we looked at body length, body weight, number of eggs laid, and longevity. And essentially the main takeaways here are that the Drosophila reared with royal jelly medium had an increase in body size, eggs laid, and extended lifespan while experiencing a shortened developmental time. And so the other takeaway is that royal actin increased body size, cell size, and eggs laid while extending lifespan and shortening developmental time. And so this was not seen in the 450 kilodalton protein or the casein or just the royal jelly denatured by itself. And so overall, fresh royal jelly led genetically identical fly larvae to develop into adult individuals with phenotypes similar to queen bees, showing that Drosophila could be used as a model organism for genetic analysis of caste differentiation. Moving forward, going back to honeybees, we're looking now at the suppression of queen differentiation with honeybees using RNAi. And essentially, RNAi is a way of silencing genes by mRNA degradation. And so it's a way of gene knockout. And so we're going to actually look at the pathways involved in caste differentiation in these honeybee larvae and reared with the suppression of either their insulin receptors, INR, their epidermal growth factor receptor, EGFR, or S6K, which we'll touch on in a bit. And so in this figure, we have five groups of larvae, one reared with royal jelly that's stored for that period, and then 4 with fresh royal jelly, just with suppression through RNAi. And so here we knock out the GFP, or green fluorescent protein, which simply acts as a control of the RNAi mechanism. And then we either knock out the insulin receptor, like I said, or the epidermal growth factor, and then also S6K, which is essentially a kinase that regulates cell growth proliferation through protein synthesis. And once again, we look to measure developmental time, weight at adult emergence, and ovary size. And so the main takeaways are that the knockdown of INR did not affect adult size, developmental time, or ovary size. But in contrast, EGFR knockout did reduce adult size and ovary size and prolong developmental time. And so this indicates that the activation of EGFR by royal actin is involved in caste differentiation. We'll also note here that suppression of the S6K with RNAi did not have an effect on developmental time or ovary size, but did on the final adult size, as we can see through these figures. And so moving forward with the conclusion, royal actin acts on epidermal growth factor, or EGFR, in the honeybee to induce queen differentiation, shown through increased body size and ovary development, as well as shortened development time in honeybees, all characteristic of queens. So this means that EGFR signaling has a very important role in growth regulation. Another takeaway is that administration and overexpression of royal actin in Drosophila cause morphological and physiological changes resembling the queen bees, once again showing caste differentiation. And we can also show that royal actin induced prolonged longevity in the Drosophila and the Apis mellifera, showing that it might have an important role in the prolongation of longevity in queens. Now, finally, showing the significance, this data is important because it allows us to understand the relationship between royal jelly and caste differentiation, effectively bridging the two, which will equip beekeepers to produce royal jelly with the proper concentrations of royal actin in order to maintain queen populations and overall hive health. This is important worldwide, as it's very important to promote the survival growth of honeybee hive populations overall through adequate populations of queens. Thank you very much, and gig 'em.